Hello, everyone. Welcome to Meds.Concepts. In today's lecture on basics of transcription. Transcription is one of the most fundamental processes in molecular biology, where genetic information stored in DNA is converted into RNA. This process forms the first step of gene expression and ultimately controls protein synthesis. In this lecture, we will discuss the transcription unit, coding and template strands, RNA polymerase, promoters, monocistronic and polycistronic mRNA, and important transcription-related concepts. Central dogma and overview. The flow of genetic information in biological systems is explained by the central dogma proposed by Francis Crick. According to the central dogma, DNA is transcribed into RNA, and RNA is translated into proteins. Thus, genetic information generally flows in the direction DNA to RNA to protein. Replication duplicates DNA. Transcription synthesize RNA from DNA. Translation synthesize proteins using mRNA. In some viruses, reverse transcription also occurs where RNA acts as a template for DNA synthesis. Transcription therefore serves as the bridge between stored genetic information and functional gene expression. What is transcription? Transcription is the process of RNA synthesis using DNA as a template. RNA polymerase reads one strand of DNA and synthesizes a complementary RNA molecule. Several important features characterize transcription. First, it is template dependent. Second, RNA synthesis occurs in the 5' to 3' direction. Third, transcription does not require a primer unlike DNA replication. RNA polymerase uses ribonucleoside triphosphates or rNTPs. ATP, GTP, CTP, and UTP. Only one strand of DNA acts as the template strand for a particular gene. The synthesized RNA is complementary and antiparallel to the template strand. Requirements for transcription. Several components are required for transcription. These include DNA template, RNA polymerase, ribonucleotides, Mg2+ ions, and promoter sequences. RNA polymerase catalyzes phosphodiester bond formation between ribonucleotides. Mg2+ ions are essential cofactors for catalytic activity. An important distinction between DNA polymerase and RNA polymerase is that RNA polymerase does not require a primer. DNA polymerase uses deoxyribonucleotides or DNTPs, whereas RNA polymerase uses ribonucleotides or rNTPs. Transcription unit. A transcription unit is the region of DNA that is transcribed into RNA. It consists of three major components: promoter, coding region, and terminator. The promoter is the DNA sequence where RNA polymerase binds. The coding region contains the information copied into RNA. The terminator signals the end of transcription. The transcription start site is designated as +1. Nucleotides before +1 are assigned negative numbers, whereas nucleotides after +1 are assigned positive numbers. Upstream and downstream regions. In transcription terminology, DNA regions are described relative to the transcription start site. Sequences located before the +1 position are called upstream regions and are assigned negative numbering. These regions usually contain promoter elements. Sequences located after the +1 position are called downstream regions and are assigned positive numbering. RNA polymerase moves downstream while synthesizing RNA in the 5' to 3' direction. Coding strand versus template strand. DNA contains two antiparallel strands. During transcription, only one strand serves as the template strand. RNA polymerase reads the template strand in the 3' to 5' direction and synthesizes RNA in the 5' to 3' direction. The RNA sequence is complementary to the template strand. The second DNA strand is called the coding strand or non-template strand. Its sequence is identical to RNA except that thymine in DNA is replaced by uracil in RNA. For example, if the coding strand contains ATGC, the RNA sequence will contain AUGC, exons, introns, and intergenic DNA. In eukaryotes, genes are often interrupted by non-coding sequences called introns. The coding regions that remain in mature RNA are called exons. During RNA processing, introns are removed through splicing and exons are joined together. DNA sequences located between genes are called intergenic DNA. Not all non-coding DNA is useless. Many non-coding regions regulate transcription, chromatin structure, and genome organization. Consensus sequence. A consensus sequence is an idealized DNA sequence representing the most common nucleotide present at each position among similar sequences. Consensus sequences are important for promoter recognition by RNA polymerase and transcription factors. In prokaryotes, an important promoter consensus sequence is the Pribnow box located around the -10 region. Its consensus sequence is generally TATAAT. In eukaryotes, the TATA box is an important promoter element located approximately 25 nucleotides upstream of the transcription start site. Consensus sequences help transcription machinery identify promoter regions efficiently. Monocistronic and polycistronic mRNA. Messenger RNA molecules may be monocistronic or polycistronic. Monocistronic mRNA contains one open reading frame and encodes a single protein. This type is common in eukaryotes. Polycistronic mRNA contains multiple open reading frames and encodes several proteins. This arrangement is common in prokaryotes where functionally related genes are organized into operons. Thus, a single transcript can produce multiple proteins. Operon and Transcription Unit. An operon is a group of genes transcribed together under the control of a single promoter. Operons are characteristic features of prokaryotic gene organization. A typical operon contains promoter, operator, and structural genes. The promoter binds RNA polymerase. The operator acts as a regulatory site for repressors. Structural genes encode proteins involved in related functions. The lac operon is a classic example of a prokaryotic operon. Transcription of an operon produces a single polycistronic mRNA. RNA polymerase types. Different organisms contain different types of RNA polymerase. Prokaryotes possess a single RNA polymerase responsible for synthesis of all RNA types. In eukaryotes, 3 major nuclear RNA polymerases are present. RNA polymerase I synthesize most ribosomal RNAs. RNA polymerase II synthesize messenger RNA and some small nuclear RNAs. RNA polymerase III synthesize transfer RNA and 5S ribosomal RNA. Mitochondria and chloroplasts also possess their own RNA polymerase because these organelles contain their own genomes. RNA-dependent RNA polymerase. RNA-dependent RNA polymerase is also known as RNA replicase. Unlike normal RNA polymerase, it uses RNA rather than DNA as the template. This enzyme is mainly found in RNA viruses. It synthesizes RNA in the 5' to 3' direction. Examples include influenza virus, bacteriophage cubita, and other RNA viruses. These polymerases specifically replicate viral RNA genomes and generally do not replicate host cellular RNAs. Features of DNA-dependent RNA polymerase. DNA-dependent RNA polymerase possesses several important characteristics. It is template-dependent and synthesizes RNA complementary to DNA. It does not require a primer for initiation. Mg2+ ions are essential for catalytic activity. RNA polymerase is slower than DNA polymerase. The approximate transcription rate is around 40 nucleotides per second, whereas DNA polymerase synthesizes nearly 1000 nucleotides per second. Unlike DNA polymerase, RNA polymerase lacks strong proofreading activity and therefore has lower fidelity. However, transcription errors are usually less harmful because RNA molecules are temporary. Let us quickly revise the important concepts from today's lecture. Transcription synthesizes RNA from DNA. RNA synthesis occurs in the 5' to 3' direction. RNA polymerase does not require a primer. RNA is complementary to the template strand. The coding strand has the same sequence as RNA except T is replaced by U. Prokaryotes poses a single RNA polymerase. RNA polymerase to synthesize mRNA in eukaryotes. Polycistronic mRNA is common in prokaryotes. These concepts are the foundation for advanced transcription mechanisms. In the next video we will discuss transcription in prokaryotes. Thank you. If you found this video helpful, please like, subscribe and share it with your friends.